Welcome to the Everything Epigenetics podcast, where we discuss DNA regulation and the insights it can tell you about your health. I'm Hannah Wendt, and I'm the founder of Everything Epigenetics. Today, my guest is Dr. Varun Dwaraka. I work with Varun as he's the head of Bioinformatics at True Diagnostic, and I've known him for about three and a half years now, and we have become good friends over that time. This is a really cool conversation because I'm not always up to date on all of the bioinformatics happenings at my company. So we really dive into his background and how he got involved in epigenetics and what he really studied during his graduate school years. But we further get into, what's an epigenetic clock? I've previously discussed the generations of epigenetic clocks on my podcast, but really, how do we make these clocks? What are the best bioinformatic practices when we're creating these types of clocks? And we talk about reproducibility, using principal component analyses, corrected algorithms, making sure these clocks are related to clinical outcomes and have association to disease. And we even talk the validation of these clocks and what that looks like, and that it's important to use the specific tissue type the clock is built in. My favorite part of the discussion dives into these new EBPs, or this idea of epigenetic biomarker proxies. And we actually talk about the difference between an EBP and an MRS, or a methylation risk score. We end with what other areas of discovery really excite Varun and the specific projects that he's working on at the moment. A little bit of background information for my guest. Like I mentioned, he is the head of bioinformatics at True Diagnostic, an aging and longevity investigator that specializes in epigenetics and bioinformatics. He's co-authored numerous publications relating to genetics, epigenetics, DNA methylation, and tissue regeneration. He currently serves as a 2023 Foresight Fellow in Biotechnology and Health Expansion, awarded by the Foresight Institute. Dr. Dwaraka is passionate about implementing machine learning methods to advance predictive medicine, identify novel biomarkers, and create algorithms to better understand the biology of aging. Now for my guest, Varun Dwaraka. Welcome to the Everything Epigenetics podcast, Varun. I'm excited to chat with you today.
Yeah, this is awesome. And by the way, can I pay you a compliment before we start?
Let's do it.
Because this podcast has actually been one of those podcasts I've been listening to, mainly because I commend you for how you're distilling a lot of the epigenetics world into something that is approachable. And so hats off to you.
Thanks, Varun. I appreciate it, kicking it off with a compliment. Well, cool. No, I'm super excited for this episode. As I alluded to in the introduction, Varun and I work together. We first met, I don't know, maybe 2 or 3 years ago now, which seems crazy.
It's coming on to 3, yeah, about 3 years ago.
Yeah, so I'm excited. I'm excited to dig a little bit deeper. We're good friends as well. We get to see each other in office every day and work on some really cool stuff. So, when we first met, and it was, like I said, 3 years ago. Now you lead the bioinformatics team at True Diagnostic. I think you are the most knowledgeable when it comes to biomarker discovery using machine learning, AI, really large data sets through the lens of epigenetics, and creating all these different predictors of health using that data. And we'll talk into exactly what that means here during the rest of the podcast. But tell us a little bit more about how you got here and how you ended up in Lexington, Kentucky too.
Sure, yeah. Well, thank you, first of all, for mentioning that. So I guess, like, starting off, I grew up in California in the San Francisco Bay Area. And so, like, when I wanted to kind of pursue what I wanted to do in the future, I kind of naturally gravitated a little bit towards biology, even though I was a musician growing up, and that's kind of what I wanted to do. And how I ended up in Kentucky is that actually my undergrad institution, I actually went to University of California, Santa Cruz, and that actually started this whole bit on bioinformatics. So I didn't start in epigenetics. I wanted to understand how we could utilize computation and technology to better understand biological big data. Essentially, all the data that's coming out of these sequencers, these A's, G's, C's, and T's, and also the marks that are around it, how does that relate back to human health? Not only just human health, just biology in general. And so When I went to UC Santa Cruz, I worked at the UCSC Genome Browser, got to know a little bit more of that. And so when I got to Kentucky, I specifically sought out a professor, Dr. Randall Voss at the Department of Biology, to work on salamanders, on axolotls of all things, because they had this insane ability to repair and regenerate whole tissues and structures. And so I studied tissue regeneration, and one of the things that I focused on was how are the genes being activated and also deactivated during this process. And one of the things is when you start to look at, genes in terms of a circuit board, you can kind of understand, okay, these genes are being turned on, so the switches are on. These genes are turned off, so these switches are off. I started to navigate towards, okay, well, what's causing that switch? And it kind of ended up on epigenetics. And so I spent actually a large portion of my final chapter utilizing a lot of DNA methylation, specifically DNA methylation techniques to kind of address those questions.
And what year were you in Randall Voss's lab? When was that?
It was 2015 to 2021.
Okay, so that's so weird because I'm actually just putting this together for the first time. I was at UK from 2015 to 2019, and like I remember thinking of looking into Dr. Randall Voss's lab because he had the cute little salamanders and the axolotl and being able to study those for tissue regeneration. Like I remember going and trying to find a PI in someone's lab that I could be a part of as an undergrad. So like I was in that same biology chemistry department, and like I'm sure our paths crossed, but I've actually like never thought that out loud. So that's interesting.
Fun fact, we were in the same department at University of Kentucky when I was a grad student. I think you were a undergrad at the
undergrad.
time? Yeah.
Well, super cool. And like I said, I'm sure we crossed paths. If not, then eventually at some point, which of course we did at True Diagnostic. And before True Diagnostic, I didn't really know much, if anything, about epigenetics. I get that all the time. Like, how do you have all of this knowledge? And it makes me giggle because it's really all self-taught. Like I have really good teachers like you and everyone else in the company and all of the academic and institutional partners that we work with. So digging into this literature and again, learning from my surroundings is really the way we've been grinding at TrueD. But you mentioned, previously that a significant portion of your dissertation covered epigenetics, specifically looking at that histone methylation changes and DNA methylation changes in salamander tissue regeneration. So not necessarily human data, but there's definitely going to be, that overlap. And what I'm really getting at here is, epigenetics has exploded dramatically over the past decade, even over just the past couple of years. So, why do you really believe this field has been rapidly exploding, and like why do you think studying epigenetics and these changes is important, broadly speaking, broad terms?
I think I think it's because of that same experience that I felt, because I think in the later part of, let's say, the 2010s, a lot of people were doing like RNA sequencing, so looking really at how genes are being expressed and de-expressed and how these circuits are being there. I think that a lot of work is still continuing on, but I think we're at this point of saturation where, okay, we know that just figure out some kind of disease. We know that these genes are expressed in that disease. The natural question really comes on to what is upstream of that, what is contributing to that overall gene expression pattern. And if you kind of go back into biology 101, there's this whole concept that I always talk about called central dogma, where DNA turns into RNA, RNA turns into protein, you have transcription, translation, and epigenetics kind of serves as the upstream of that transcription. So, when we talk about epigenetics, I have to say you are not only just discussing DNA methylation, but we're also mentioning non-coding RNAs, histone modifications, and all that. But DNA methylation kind of comes into play because that is that is one of those marks that is readily associated with a lot of environmental factors. And so when you're starting to understand in terms of human disease what that upstream factor could be, there seems to be a lot of literature, there actually is, not seems, there is a lot of literature that suggests that DNA methylation might be the specific epigenetic mark that really, kind of goes downstream into the central dogma. And so that was,
Yeah.
I think that's why epigenetics and particularly DNA methylation in the context of human health has really exploded.
Yeah, definitely. And I'm obviously not going to throw them out there or, mention any names, but I think too, just as researchers in this field, we're seeing a lot of people in these really large labs who were betting on transcriptomics previously, now betting on epigenomics and, quite literally transferring all of their funding, all of their sources to now studying kind of a different omic layer. So we're seeing epigenetics, like I mentioned, just explode.
Yeah.
And we'll continue to. I have no doubt in my mind. I mean, there's a new company created every single day, a new lab created every single day that's actually trying to understand what these insights mean, or even epigenetic reprogramming and trying to set these ages of cells, so to speak, back to zero to figure out how we can do that on a larger scale.
Yeah, agreed. And I think that's what's the beauty of it all. It's that, no one stumbles into this. I stumbled onto like as an accident. I vividly remember being in my advisor's room, office room, which was actually it's funny. He used to work with the salamanders. Like he had his own office, nice little office, but he would always stay in the stock center. I remember coming into there and I was like, look, I naturally, like, what is causing all of these switches to be the way that they are? And he was like, well, why don't you find out? Start with epigenetics. And he literally gave me a data set and was like, just learn it. There is a textbook I have on epigenetics. I maybe looked at a few pages, but a lot of the learning, and I think that you feel the same way, people in our company feel the same way, and even other people in the field feel the same way. You have to go to the primary literature because this is it's not a nascent field, because Adrian Bird back, I think in 1967, 60s and 50s, really spearheaded a lot of the understanding of these DNA methylation marks. But I think moving forward, I think the boom of next gen sequencing actually has, made it a super exciting and field, which is now rapidly evolving. So I think the primary literature is really where you're going to learn a lot more of it
Yeah.
in the context of today.
Definitely. And I would not know this answer off the top of my head, so no pressure if you don't know it. Varun, do you know who wrote that epigenetics textbook by chance?
I believe it is. I have it. I think it's like C. Alice. I think it's Alice. Last name is Alice.
Interesting. Okay, yeah. Well,
Yeah,
well,
David
I'm
Alice.
just like
David
interested
Alice.
to see
Yeah.
like how long ago it was written and like what's included in that. We need to write a book on epigenetics. It's like I don't even know where you would start. But it would be like so introductory level. I feel like yeah, and you could go into,
Yeah.
any and all different areas of health. But like many of you know, at my company, True Diagnostic, we really use epigenetics. Well, it started out using epigenetics to measure aging because that's where we can actually have the largest impact. So this is a really good segue into what I want to talk to you about next, Varun. Aging's that number one risk factor for all-cause mortality, morbidity. So not only is there a massive healthcare impact that we're talking about here, there's a huge financial aspect too. And for those so some people listening may have seen Dr. David Sinclair's paper he wrote on this, in which the economic value of targeted aging is just huge. Yeah. So, for example, if we increase life expectancy just by 1 year, it's worth $38 trillion. And the US debt is around $34 trillion, right? And it keeps growing and growing. So this is usually a number, this biological aging I'm talking about here, in like a clock format. And I don't know that I've ever actually, even defined what an epigenetic clock is, or, more with your skill set, Varun, on how these are actually created and made and, what really defines a clock. So can you even just in simple terms explain like what's an epigenetic clock, or what is a clock? What does that mean?
Sure. Yeah. A clock is kind of derived from this idea of a biological clock. And so based on your date of birth, the time that you're born, you can have something called a chronological age clock, where it's based on the time you were born and the date that you were assigned now, essentially, or that you have now. A biological clock is essentially the age in which your body represents. So you might be 24, but you have the body of a 22-year-old, or you're 34 and you have a body of a 24-year-old because you take really good care of yourself. This goes back into how are you estimating that biological age? You can estimate biological age multiple ways. You could use peptides, you could use proteins, you could use clinical markers. And so an epigenetic clock is utilizing epigenetic markers. Specifically in this case, we're talking about DNA methylation, but I will have to say there are transcriptomic clocks and histone modification based clocks as well. But when we talk about epigenetic clocks, largely we're focusing on DNA methylation markers. It's essentially the orientation or the methylation of the DNA methylation sites contributing to that biological age calculation from a numbers perspective, essentially. So an epigenetic age clock is your biological age based on your DNA methylation, essentially.
Yeah, and there's different types of those as well, right? So I think it's essential to even just define a clock. Like you said, you can create a clock with many different omics or many different biomarkers. These epigenetic clocks, of course, are going to be using DNA methylation data or epigenetic data. And I think I have previously explained the difference between the first, second, and third generations, right? First generation is just using chronological age information along with DNA methylation. Second generation is going to be better because it's using not only the DNA methylation, but other underlying biomarkers. So it's going to be a better predictor. And then when we think third generation, remember, we think of that Dunedin pace algorithm, more of this longitudinal analyses. So,
and we love that Dunedin pace algorithm.
Yes, we love the Dunedin pace algorithm. It's very unique, and it's still my favorite one to talk about to this day. And this is why I wanted to have you on, Varun, with your expertise in bioinformatics, statistics. What are the best bioinformatic practices when you're actually creating these types of clocks? And feel free to really go into detail here as well.
Absolutely. I think based on the experiences we've had, especially at True Diagnostic, and also just vetting a lot of these clocks, the first one I can think of is the reproducibility. So I think there was a paper by Dr. Higgins Chen with Morgan Levine when she was in Yale University, where they actually looked at a lot of the clocks that are currently out there. So those first generation, second generation, third generation clocks. And what they started to do notice is that the reproducibility, that like you take the same biological sample and you quantify the methylation, the variability in those outputs weren't that great. You'd have like skews of like 3 to 4 years difference from the same exact output. And so they started to utilize this metric called ICCs, intra-class correlations, which is essentially a correlation between how reproducible or how correlated are the same values from repeat tests. And so when we start to address the first thing to address these clocks is how reproducible and how correlated are those repeat measures. And one of the reasons why we always tout Dunedin pace and additional clocks as well. I think the reason why I brought up Dr. Higgins Chen is because they actually recreated some of those first and second generation clocks using this principal component technique. The main reason was to improve this ICC, this intra-class correlation or reproducibility of these epigenetic clocks. So that's number one.
And can you just very briefly to define what principal component analysis means?
Yeah. So principal components it's a statistical measure, a method where rather than looking at individual CPGs, you can clump multiple CPGs or group multiple CPGs that are numerically very similar. And so what this allows is reliability. Now, when you're doing a lot of these non-PC clocks or principal component clocks, you're estimating individual CPGs. That's great. But the problem is actually when you run, let's say you run a sample through, this methylation quantification. If there's some technical error in one CPG by random chance that is highly important for the quantification of that clock, then you skew the entire data set. Right? That value doesn't actually work out too well. And so why this principal component was a really neat method is because now you're not privy to just one CPG, because even if that one CPG fails, all of his buddies are still there. All of the other all of the other CPGs that are part of that one CPG are still hopefully, unless it's some major issue, are still relevant and they're still, being quantified in a very positive way. And so that allows for that reproducibility to actually occur.
I like that. It's the principal, like all your buddies, your little CPGs. I'll start describing it that way. And when Varun's saying CPG, he just means that DNA methylation
Methylation.
marker, that cytosine, phosphate, and guanine, if we want to really start and define some of these terms. But that's a good point. I usually explain it to people as well. Isn't it, when you have the principal component analyses algorithms, isn't that like thousands and thousands of CPGs included in those actual algorithms?
That's precisely correct. Yeah. A lot of
Okay.
these non-PC or principal component-based clocks will only maybe measure about 350, let's say 3, according to the Horvath clock. But when you start to look into principal components, you can actually incorporate many more CPGs just based on how the methodology kind of works out. So you're actually imparting more of the signal.
Yeah, they're kind of like leaning on each other. I almost think of it as, I just think of it as like adding another layer of like QA, QC almost.
Absolutely.
I don't know. It's the kind of most simple way I've felt like I could describe it to healthcare providers.
But I would agree with that. I think
patients.
you are adding an additional QA, QC to it. So I don't think you're off base there.
Yeah. All right. What else? So you have, reproducibility, adding in these principal component analyses clock, which, when you're adding those on top of the baseline algorithm, it accounts for a lot of those differences. So that's really important. Maybe the next, top 2 or 3 best bioinformatics.
The next thing that I could think of is honestly, again, if you want something that's clinically reliable, you want to also know like what its association is to clinical outcome. And so this is, I think, where it really separates out some of the first generation clocks to, let's say, the second or third generation clocks, is the association to disease. I will say this: the first generation clocks actually have associations to diseases, right? Like the Horvath clock actually shows significant association to cancer risk. And so depending on what the question is or what the clinical outcome that you're really interested in will dictate which of these clocks are there. However, once you start to incorporate more of the biological, let's say, signals, like for example, from phenoage, which actually is trained not to chronological age like these first generation clocks, but more to a phenotypic score that is based on biological markers, what you're actually doing is imparting a lot more biological input. So you're not just basing it again on something that is, constructed by chronological age. You're constructing it more on the biology. And what you start to notice is that when you, let's say, associate to time till death, let's say you want this measure to reflect how long a person might have to live, mortality. What you start to notice is that these biologically derived clocks show stronger associations to overall outcome, mortality outcome, than, let's say, these chronologically trained algorithms. And this is why that narrative of the second generation being better kind of comes into play. Is first generation does its job, and it's still a relatively, it's a clinical output that's relatively positive. But when we want to increase the breadth of that clinical utilization, that's where the second and third generation really come into play. And I will say one thing too is that the third generation, I know that there's a kind of a differentiation. Some people call the Dunedin pace the third generation, and there's like a mammalian clock that is now third generation. The way that I'm using it, third generation, I'm representing Dunedin pace. So just wanted to make sure that the listeners are clear about that.
Got it. Yeah, the language is important here because there's a lot of unknown terms or terms that have been defined in a multitude of ways. So you're right. First generation, I like how you said it, does its job. Second generation, they're probably better, right? Well, we know they're better because they're more predictive of outcomes of all-cause mortality and morbidity. So that means if you're testing a second generation clock and your biological age you get back is actually older than your chronological age, you are at more increased risk for all-cause mortality and morbidity. It's going to be more predictive.
Yeah.
And same is true of the opposite. If you test and your biological age is younger than your chronological age, great, even better. You're at even much more of a reduced risk of all-cause mortality and morbidity.
Yeah. And actually, it's funny because today, just today, we put out a paper in Nature Aging, which was talking about stochastic clocks. Now, the stochasticity of it all is, I think this is a term that might be, like, it was hard for me to understand initially when we were kind of just even talking about the study. But this idea is that there are two aspects of aging that I think people need to consider. What is your stochastic aging and what is your dynamic aging? Your stochastic aging is the aging that you're accruing throughout your life. So this is, as you exist in your environment, you are garnering changes in your biology, even from the molecular side. In terms of DNA methylation, you're accumulating DNA methylation as you just live. And so that's going to be your stochastic aging. These first generation clocks do really well in capturing that stochastic age. And that's what this paper kind of talks about. However, there's also a quasi-stochastic or more dynamic aging that's also being captured. And what we show is that, these biologically derived clocks, namely PhenoAge, actually does a really good job in capturing that dynamic aging, which is more associated to, in a short amount of time, that you might have a rapid increase or decrease in that age, which might be associated to disease onset or, let's say, improvements in overall health outcomes. And so that paper was really, we were really thankful to work with Dr. Teschendorf and his group, and also Dr. Jessica Lasky-Sue. But I think that really starts to show the importance of these different generations, and also kind of hone in on, when you're trying to create something that's clinically relevant, what you should be really focusing on.
Yeah, and I think, that data suggests that, like you said, the first generation clocks, which are trained to predict chronological age, they have less utility in predicting outcomes of aging than clocks like the second generation that are trained on other phenotypes of aging or other biomarkers. So this is just another point to prove that we need to be talking about this, especially in epigenetic clock development for future development and refinement of these markers, right? This really guides kind of where we want to go next in the field. There has to be almost like some type of an agreement when we're creating these clocks. And again, that's why I like to ask, what are these best practices? Because then we may be able to come to an agreement and follow them or train them in the same way. Yeah. So we can really start to get more of those interventional trials and see what's actually moving the clocks in terms of increasing and more relationship to disease, or hopefully, decreasing. So also, Varun, can you just, like, in one sentence define like stochastic aging versus dynamic aging again, just so I can make sure like we have that correct? Because to be quite honest, I haven't really read the paper fully.
That's all right. Yeah.
I'm still trying to wrap my head around this idea of aging.
Yeah. So stochastic represents just random chance of accumulation of any kind of signal. So stochastic aging is the aging marks that you accrue randomly. Dynamic aging, or the quasi-stochastic, as we kind of call it, is associated with some actual reason for that aging to occur. So this could be disease onset. This could be, exposure to environmental cues, any kind of external factor. That's not random.
Thank you. That's perfect. And they're both changeable though, right?
Yeah. I mean, the idea of stochastic aging is that it accrues just naturally. And so I can't say much of how much of stochastic change or stochastic aging can be changed. Definitely the dynamic can, though.
Right. Okay. Got it. That makes a little bit more sense. Again, first generation clock measuring more chronological age. It's like natural aging. There's random chances that you're gaining, methylation and that's affecting your overall aging. This dynamic aging, there's some actual underlying reason, whether it's like a biomarker change or whether it's like a disease type of change that it has in terms of affecting the body.
Yep.
Awesome. Cool. Thanks for bringing that paper up. I know that's a really exciting one and was just published today on the date that we're recording it, and I will definitely link it out. All right. So we talked about reproducibility, including the principal component analyses algorithms. Number 2, these epigenetic clocks have to be related to clinical outcomes. They have to have an association to disease. Second, third generation look better than first generation. We now have even a better argument behind that with this stochastic aging and this dynamic aging. Yep. If you had to pick one more, because there's a lot of other things we need to get to, what's another best bioinformatic practice that you would include?
I don't know if this is bioinformatic, but I would almost say this is more scientific and kind of goes with the second one. It's validation. I think these clocks need to be vetted, not just based on, white papers or like an N of one style, clinical cases, but really in these randomized control trials, in these more large population kind of not only meta-analyses, but just interventions. Because what I think that affords is no one clock is built the same. It's inherent. Like when you start to calculate a lot of these outputs, you'll notice that the CPGs that are shared is actually very minuscule, really. And so naturally, even from a numbers perspective, the numbers are going to be different. This is why the clocks, and I know, sorry, can I go a little bit off the rails here? I understand that it's really disheartening to see multiple numbers not agree with each other, but people need to understand these clocks are built for specific uses. And so if this clock is showing you as older, or in this clock is showing you as younger, there's a reason for that, because it's capturing a different part of aging that is saying you are older, or a different part of aging that is younger. And so this is where, and the reason why I'm going off the rails here is because this is where you need to go back, anyone you work with, in order to interpret these epigenetic outputs, you have to go back to the validated interventions. This clock is reduced in this context. Are you doing that? That could be a easy way to kind of start that conversation. And so
Yeah.
I think that validated interventions, and I think the more clinical trials that these clocks are part of, will then just open our eyes to where does it work, where doesn't it? And I think that that'll allow us to know more about where to utilize it too.
Yeah. Awesome point, and a good one to end on in terms of, I know there's a ton of other things we can add onto this list, but number 3, validation. And my follow-up is going to be, well, Varun, what do you consider validation? But really, these randomized control trials, more clinical trials, what can you do to really move these and change these? Did it work previously in animal models? Does it work to really affect our underlying biology?
Yeah.
That one speaks to me just because I'm really helping people try and interpret these results on a daily basis. And, I understand where the frustration and confusion may come from,
and I know you said something about this being the last one. And you touched on it there. You also have to know which tissue type it was built on, because that's, you don't want to use something that was built for saliva on blood and vice versa. So I wanted to make sure we added that too.
Yeah. Fourth one, tissue type.
Yeah.
Tissue type has to be consistent, and we need to address that, and we need to look at that. So no, that's great. That gives us a really good baseline on, hey, what are we actually looking at? And, it's still way too early. We have a couple randomized control trials and we're getting really closer. We have a lot of internal data at True Diagnostic that we don't do a great job of sharing, but we will be getting better here soon. What's, really exciting though is, that we are still so early. This podcast, it's a lot of research. It's a lot of far out there ideas almost. And I do imagine one day, we look at this 10 years from now and we just laugh and say, little did we know. We thought we knew so much then. There will be so much that we start to uncover. So I'm just super excited about the possibilities there.
I am too. I think that's the reason why you go into science, or you do any of this kind of scientific work, right? And I think that's one thing about where we're at True Diagnostic, is that we understand, in order to better provide for individuals, we have to understand the science first. We can't provide something without knowing how it was made or how it's operating. And I think that with that, we feel the same way as actually a lot of the researchers do. I look up to a lot of the Ricardo Marionis of the world, Steve Horvaths, Morgan Levine's, Daniel Belsky's, Terry Moffat's, Avshalom Caspi. There are, and if I didn't mention any names, I apologize. But we look up to these individuals because I think it's a give and take. We're learning a lot of things we want to put out to the world, but we're also learning a lot from other individuals that we're trying to distill and bring out to the people that are here at True Diagnostic, and also read our papers and things like that. So history has taught me that whatever I know now will be changed in the future.
Of course.
So,
and I think at least you know that, you already know more than a ton of people. If you're open to that and you just accept that idea, you're already more than halfway there. I think that's science for you.
And
right?
I would really want people to also feel that way too. I think the people that are listening to this podcast, like I would hope that they kind of maintain that malleability, that neuroplasticity, if you will, to kind of do that as well.
Sure. No, good point. Good point. Okay, this is, I know I keep saying, I feel like every time I bring up a new question or topic, like this is the most important part or what I'm most excited to ask about. But this one really is probably the most exciting thing I want to discuss today. And it's just the other use cases for epigenetics. So I used to describe like my company as, oh, we have, the best biological age testing. And that's still how most people find us. And again, where we can really make the biggest impact. But that's old news to me. It doesn't get me excited. It doesn't get me out of bed every day. It kind of bores me a little because there is this new idea of using epigenetics to report out other outcomes.
Yep.
And that is going to be what is called an EBP, or epigenetic biomarker proxy. So it's a new term. It's a completely new idea. So Varun, you've been leading this at True Diagnostic. Can you just define what is an EBP?
EBP was a term that we kind of coined in the context of what we're doing. So the idea of an EBP is that we're using DNA methylation data to essentially impute or estimate values for proteins, for metabolites, and for clinical biomarkers that you don't actually need to take, that you don't actually have to do traditionally. And so using the same data set, you can get estimates of those values. This really came kind of on the heels of a lot of the work that Riccardo Marioni has done in the past, where I think he coined his called Episcores, where he was able to, or him and his group were able to utilize DNA methylation to estimate proteomics. Now, how does this work? I think it goes back to the central dogma. We know that epigenetics is connected to DNA. DNA then goes to RNA. RNA then goes to protein. So they are relatively linked. There is some shared signal that is going from the beginning to the end. And therefore, and then also with that protein, that elicits a phenotypic or physiological response, which is captured by those clinical tests. It's under that framework that's why the logic of the EBPs was kind of generated. And so how we created this was that we actually worked with Harvard University Brigham's biobank, specifically Dr. Jessica Lasky-Sue's group, where we collected samples from their biobank, about 5,000 or so, and then calculated or quantified their DNA methylation, their metabolites of a subset, the proteomics of a subset, and the clinical biomarkers of a subset. And throughout a lot of machine learning modeling and a lot of that, we were able to create models where you identify which CPGs are most predictive of that final outcome. And so the reason why we did this initially was for aging. We wanted to build a multi-omic clock, which took all of these values and kind of coalesced it or condensed it into a single measure. But what we started to realize is that even though we created a composite, a lot of people are really interested in what is contributing to that composite score. So how is this EBP actually affecting it? Because sure, you might be 76 if like chronologically, but you're 78 based on your omic age. So then naturally it's like, okay, well, what's contributing to that? Then you can start to go into the EBPs because they are part of that calculation. And then say, oh, it's that CRP that's really, not that great. It might show a percentile that's like 90th percentile. We can get to that in a little bit more details, but that one is actually contributing, is really high and therefore contributing to the overall omic age. Your doctor, your physician will likely know or understand how that CRP is being affecting, or how to reduce that maybe. And then you start to actually gamify, know exactly where to go in a more personalized approach.
Yeah, that's massive. I mean, everyone, should be on the edge of their seats right now, right? This is huge. The implication of these epigenetic biomarker proxies and giving you guidance on what you need to work on to lower your biological aging is massive. So right now, there is reporting that tells you what they are and what you need to work on to lower biological aging. But I know furthermore, we're actually reporting these out on a 1-to-1 ratio. And, I get my bloods drawn every 6 months. Like I'm sick of going to LabCorp. I'm sick of going to Quest. I'm sick of fasting and not eating until I get my bloods drawn and, trying not to exercise in the morning and trying to control for all of these other external variables. But what if you could just be sent a kit, you're sitting at home, you take out the blood spot card, you prick your finger with the lancet, you fill up one little circle that's about the size of a quarter, let it dry, and you ship it to a lab and you get results back, I don't know, in 2 to 3 weeks. And it's a very lengthy but in-depth and important report that includes aging outcomes. It includes clinical lab values. It includes protein levels, and it includes metabolites. Like, that's what we're getting at. That we're not too far away from that. And then it acts as a one-stop shop with personalized microdoses for recommended supplements.
Yeah.
So it really is insane. So aside from even those EBPs, Varun, I know you all are working on, there's like 4,000 of them that you can just like report out on from a couple drops of blood. Is that correct?
Sorry. So apologies. Can you say that one more time? Because I think that this is a really important question. Can you say that one more time?
Yeah. So, and again, like, this is me not being as involved in, the bioinformatics and the science that's even happening at True Diagnostic. So, because we're always doing so many different things, but essentially there are now these 4,000 EBPs, I believe, that we're creating with Harvard to report out on a 1-to-1 ratio. Is that correct?
So that's where we're actually going towards. And I will say this, and I want to make sure, because I definitely am hearing what people are saying. Sometimes your lab values and your EBPs may not 100%, there might be differences, right? We have been working our hardest to get them to 1-to-1, but what we also start to realize is that these EBPs are also picking up signals that are not even manifesting downstream. So meaning they're not even coming out in the clinical values, like let's say your CRP. The reason why I'm almost pivoting a little bit is because of those 4,000, we're actually running a study right now to understand why that difference is there, because previous work has shown that the CRP, for example, at the end, the clinical CRP is not necessarily as associated to age-related outcomes as the DNA methylation CRP is. So that difference there is actually attributed to more of capturing a different signal associated with aging compared to what the traditional lab values are coming out with. And so right now at True Diagnostic, we've been able to expand our list from 396 that were originally described to close to 4,000. And it's under that pretense, and why we've continued this way is because we're starting to notice that there's actually potentially more predictive power with these EBPs than there were before. So even if there's not an equal match, even though we spent a lot of time to make them 1-to-1, still there's not always 100% match, but that's actually okay. That's good, because now what you're getting is a more predictive measure for your age related outcome.
Yeah, exactly. The CRP is a really good example because it was even done by an external group. I forget who exactly. I'll link the paper. But where the DNA methylation CRP is actually more reproducible than the traditional CRP head to head, and it's actually related to brain health outcomes, where your traditional CRP is not. So it's like a 3 to 6 month running average instead of just your plasma-based biomarker at a specific point in time. So a lot of people even like to compare that DNA methylation CRP like the HbA1c of inflammation. So yeah, that's a really good example and a super exciting one. I already know like that part of the podcasting conversation will be my favorite of this entire episode, because there, again, is so many things we're finding out, but so much we don't know. And then we'll actually be able to go back and look at each individual EBP and say, hey, what is this really telling us? What is this really capturing? I mean, it's not necessarily, it's novel. It's capturing something that we probably didn't even know we should be looking at as it relates to our health, but it's probably associated to a lot of other outcomes downstream.
And this is the thing, like, obviously you can do all of this in one go. That's fantastic. But if you are really inclined to understand what your clinical outcome is, we're not stopping you from that. We're just saying that what we're essentially measuring is likely showing you a signal that is, first, clinically relevant, but two, maybe more predictive. And then if you really want to figure that out later on, you can then also still do the clinical outcomes. That's privy to you.
Yeah. Yeah, not an end all be all like replacement
Yeah.
test. That's not what we're saying. We're saying do them all together. And we'll be able to tell you a little bit more about what
But
those
you might be
actually mean
squeezing a little
soon.
bit more
So
juice with us. I'm just saying.
yeah, yeah, exactly. Now, just really quickly, we're kind of
Mhm.
getting to the end here. I know on a previous episode I talked about methylation risk
Yeah.
scores or MRSs. This was Dr. Michael Thompson out of UCLA. Um, and you all can listen to that episode if you really want to take a deep dive into his particular
He's great.
study. But Varun, just so if people are listening to these in order, like what's a methylation risk score again? And then can you just briefly explain like how does that compare to an epigenetic biomarker proxy or
Sure.
EBP?
Yeah. So essentially, the way that we trained or generated these epigenetic biomarker proxies were identifying CpGs markers that were highly associated to, let's say, a protein value. You can do the same thing directly to just disease onset. So let's say this individual has cardiovascular disease, this person doesn't. There is a statistical method, a machine learning method, where you can identify which CpGs can actually quantify or predict, the onset of that disease. And so this is kind of using Cox proportional hazard models for those statistically inclined. And ultimately, it's rather than using age as a surrogate to estimate the risk of that disease happening, which is typically what we kind of do with omic MH, is that we calculate omic MH and then, compute a disease that comes out of it, like a estimated disease outcome. Why not just go directly to the disease itself? And so it doesn't negate anything about omic MH, but what it does is that it allows a direct comparison to the disease outcome. And so that is what essentially that methylation risk score is. It's a risk of that disease or whatever that phenotype that you're looking at, or disease comorbidity whatsoever, and predicting that based on directly from the methylation marks that are quantified by DNA methylation.
Yeah, so it's your actual risk, and not to confuse listeners, but I think we were even going to call our EBPs originally MRS scores, methylation risk scores, but it just didn't feel right. It's not a risk, it's not a disease risk. So it's an epigenetic biomarker proxy, and I love it. It's an EBP. It's using epigenetics DNA methylation as the input biomarker and acting as a proxy for some other type of output. And
I remember I was at ARDD when we changed the name too. So everyone started saying EBP. I was like, what the heck is the EBP? But then I started to realize, yeah, you're absolutely right. We can't call these methylation risk scores. Like, these are proxies of that actual marker. And so I'm actually I was ecstatic when we changed it to EBPs, truth be told.
Yeah. It's cool to coin that term. Cool, Varun. Getting to the end here, what are other areas of discovery that excite you? I know there's like so many things we're working on. Is there a specific project, like I don't even know this personally about you, that you're excited about at the moment? Why you get out of bed?
I mean, I'm living the dream right now, so it's kind of hard. I was fortunate enough to be on this project looking at twins and the effect of diet on twins. But there's, I think I was speaking to Michael Corley from Cornell, and we were talking a little bit about this project. And one thing he raised up was we actually don't really know a lot about methylation patterns among twins. There have been studies, but not a lot has been done. And so I think kind of going into the whole, the twin aspect has been interesting, just because I can't think of a study design better to test how environment impacts, individuals, from a context of a genetic and just also environmental, cues. Obviously, doing that kind of an experiment is very tough. They need to adhere to a lot of compliance, as shown in this paper. But that's like one thing that would be really awesome. Number two is a lot of where we're moving towards is kind of satiating in my head this aspect of connecting molecular to output, the physiological output. How much of what we're seeing on the molecular side actually translates to clinical outcome? And I think that, really understanding that, we're starting to really scratch on that surface right now. but I think there could be so much more done. Like, for example, Whoop. Like, even from a technical side, if we can connect our methylation patterns to, let's say, Whoop outputs, what's conserved? Because I think some people would say there shouldn't be anything different, but there might be. And I think that that's really the area that I would love to kind of see, foray into. And we might.
Yeah. Yeah, I think we'll get some of that wearable data integrated for sure
Yeah.
later on. We're definitely working on some things there. So I would be excited to see those correlations or those connections too.
Yeah, and it's So like I said, it's a lot of stuff that we're working on already, but I think it's guided by this curiosity, this satiation of the question. I've been always very question oriented, and so I think that so these questions, the more papers and the more studies that we do, there's a lot more questions that come out of it. And so it may be never ending, but I think we're getting there. We're getting to some kind of end.
I think that's the important part, that we're having more questions than answers, more R&D, more discovery. So last question, Varun, this is the curveball question. If you could be any animal in the world, what would you be and why?
Oh my God. I mean, I'm going to stay away from like the usual answer, which is a dog, because I love dogs. I have Robbie is right back there somewhere, my dog. Okay. A crow. And hear me out. A crow. Crows are intelligent. They're very intelligent. They have the ability to fly, which I don't know. Flight is one of those things that's like, anytime I'm on a plane, I'm giddy. And I've been on planes for the last like 25 years. But I think it's their intelligence, their ability to utilize their environment for their own, need is incredible. And as much as
you're right, they're really smart.
As much as they're associated to like, negative contexts in a lot of, societies and religions and all that, I think from just a biological perspective, they're so intelligent and they're so crafty that, I mean,
they imitate.
And they imitate people too. Exactly. Yeah. So I'd say crow.
Yeah, they're really smart. That's interesting. Most people have like anxiety around flights and things like that too. I never knew that about you. So I'll be paying extra attention when we fly together next time.
I mean, I still hate turbulence. I absolutely hate turbulence.
Oh yeah. No one likes turbulence. Cool. We've come to this amazing podcast interview in terms of the ending. For listeners who want to connect, where can they connect with you? Where can they find you? And how can researchers access these
Absolutely,
biomarkers?
Yeah. So I think I'll start with the organization. So True Diagnostic, www.truediagnostic.com. And also on the Instagram, True Diagnostic Official. For me personally, you can find me on LinkedIn, Varun Dwaraka, VarunDwaraka. I'm on LinkedIn and Instagram as well. Same, just it's my name. And then in order to access a lot of these biomarkers, I highly implore everyone to kind of go through the papers that we've published, and then also contact me directly, Varun at truediagnostic.com, because we are more than happy to work with other researchers and other physicians on, implementing a lot of these things. Definitely Hannah has been killing it in terms of, forefronting to physicians and clients and everyone. And so I think that if there's any kind of information that is needed from a technical perspective, Hannah's perfect, but I'd be more than happy to assist in, providing additional details of the minutia of like, how any of these things. So email, Instagram, LinkedIn. Yeah. Catch me where in all of them.
All the above. Cool. We'll link them out. Well, thanks everyone again for joining us at the Everything Epigenetics podcast. And remember, you have control over your epigenetics. So tune in next time to learn more.
Thanks.
Thank Varun.
You.